Fine. So, osmosis, we discussed osmosis yesterday. Now we are going to start with the next topic. This is important. You'll get question from here. This one, this diagram, right? So, what is the diffusion or diffusion pressure deficit, right? DPD. So, diffusion pressure deficit. Please, I want your 100% focus over here, right? We need to understand what is the DPD or what is the diffusion. So, in diffusion, there are two tanks, right? This one and this one. In the both of the tank, we have sugar solution, right? Added sugar solution. And here, this sugar solution is 5 milligram, and here 1 milligram, right? So here the sugar solution is more, and here the sugar solution is less, right? So what will happen? The sugar molecule or solute molecule, they will start moving from its higher concentration towards the lower concentration, and until they achieve the equilibrium, right? So what will happen when the sugar solution will become 3 mm, like 3 milligram this point and 3 milligram this point, when both will become equal at the both of the end, means they will achieve the equilibrium. Till they achieve the equilibrium, this process will keep on going, right? This is called diffusion. And we'll know that diffusion is the movement or the movement of molecule from its higher concentration toward the lower concentration till the equilibrium is achieved. That is the diffusion, right? So now what is the diffusion pressure deficit? Can anybody tell me what is the DPD, diffusion pressure deficit? No one? Look. Now what is the diffusion pressure deficit? So what happened when these molecules, they cross the membrane? So diffusion pressure deficit, how does it take place? So this was just simply diffusion. Suppose that this is the membrane. So the molecules put pressure while diffusing molecule. When this molecule, these sugar molecule, suppose that these are the sugar molecule. These molecule put the pressure, right? These molecule put the pressure on this membrane, right? And the pressure, this pressure is called diffusion pressure, right? So here will be high diffusion pressure, high diffusion pressure, and here concentration is very low of sugar molecule, so here will be low diffusion pressure. So here, let's say here diffusion pressure is 20 mmHg, 20 mmHg, and here the diffusion pressure is 10 mmHg. Now when this diffusion pressure, this molecule will diffuse, when this diffusion process will stop, so when here become at this end it will become 15 mmHg, and here it will also become 15 mmHg. So this is the 10 mmHg. How much deficit is there? Tell me, guys. How much deficit is there? Alan, how much deficit is there to achieve the equilibrium? Mamuna, tell me, guys. Come on, guys. It's a very simple calculation. Look, here the diffusion pressure is 20 mmHg. Here the 10 mmHg. Now we know during the diffusion process, or during osmosis process, the movement of molecule will start from higher towards the lower, and when they will stop, when at the both end it becomes 15 mmHg, right? So here it's 15 mmHg. So it should be 15 mmHg. So what is the difference between the 15 and 10? There are 5 mmHg. Is it clear or not? So deficit is, how much deficit is there? Minus 5 mmHg. When this 5 mmHg, the diffusion will transfer from here to here, right? then what will happen? Then this process will stop, right? So this is called diffusion pressure deficit. The deficiency, the deficit at the lower end. Did you got it or not? Nobody is like, just tell me, guys. At least speak. Are you getting it out or not? Hello?
Yes, sir.
Getting it or not? Just need not to say barely, just yes, sir. Mamuna, did you got it? Hiba, Aksa, did you got it or not? Diffusion pressure or diffusion pressure deficit? Mamuna, hello, are you there? Hiba, are you there? Aksa and Hanifa, are you there?
Yes, sir.
So, did you got this concept or not?
Yes, sir.
Okay. So, the amount of the pressure which is deficit, right? This is called diffusion pressure deficit, right? DPD. Now, this diagram is very important. Look. Water will start moving from high osmotic pressure to low osmotic pressure, right? So, it will start moving. So, the osmotic pressure will be exerted from outside to inside the membrane, right? So, it will be exerted from outside to inside in the membrane, right? Now, what will happen when atmospheric pressure means osmotic pressure, and why osmosis is taking place? Actually, here the water concentration is high, right? Here the high water concentration, and inside the plant cell there is a low water concentration, right? So we know that the water move from its higher concentration toward the lower concentration in osmotic process. So when the water will move inside, then the water will exert pressure from outside to inside. So water will exert, or water molecule will exert or impart the pressure on the membrane of the cell. That is called osmotic pressure, OP, right? When water will go inside this vacuole. So this is a plant cell, this is cell wall, this is plasma membrane, and this is vacuole. And we know the water get collect at the vacuole, right, inside the vacuole. When this water will accumulate from the osmotic pressure, water is entering. So osmotic pressure is imparted from outside to inside. To whom? On the membrane of the plant. Then what will happen when this cell will get swell? When cell gets swell, this protoplasm will impart pressure from inside to outside. The vacuole or the protoplasm which impart pressure from inside to outside, this is called turgor pressure, right? Turgor pressure. So cell will swell, right? When you fill air in the balloon, then the air impart pressure inside the balloon. So the air which you are filling, that is a osmotic pressure. You are flowing, like you are blowing the air. The air which is getting filled, that is due to the osmotic pressure, because inside your mouth there is a high osmotic pressure of air, and inside balloon there is a low. Then you fill the after filling the air, the balloon get turgid, it will swell, right? The air inside balloon will impart the pressure, right, on the wall of balloon. That pressure will be called turgor pressure, right? Turgor pressure. Now what will happen? Now the wall of the balloon will also impart the pressure on the air, right, in opposite direction. So there will be a pressure opposite direction to the turgor pressure. This pressure is called wall pressure. And turgor pressure and wall pressure always remains the same. They remains the equal amount, right? So we have read, the swell cell is also called turgid cell, right? So turgor pressure is equal to wall pressure. Now there is a formula. What is the formula? Diffusion pressure deficit, DPD, right? What is DPD? DPD is diffusion pressure deficit is equal to osmotic pressure minus wall pressure. And wall pressure and TP are equal. So we can say osmotic pressure is dpd is equal to osmotic pressure minus turgor pressure, right? Is it clear or not? dpd is diffusion pressure deficit. Diffusion pressure deficit, right? That is the dp. And what is op? Osmotic pressure. What is wp? Wall pressure. What is tp? That is turgor. Tell me, guys, are you getting it or not? Did you got it or not? Hanifa, is it clear?
Yes, sir.
Fine. This is important concept, right? The question comes frequently from this part. Frequently question. So take the screenshot and remember this formula, right? You will get the question. Frequently, the questions are being asked from this concept. Now, the next one is the water potential. What is the water potential? This is also very important. What is the water potential? The unit to measure the water potential can be bar, Pascal, or atmospheric pressure, right? ATM. So that I have written over here, right? Water potential is represented by a term called psi. This is a symbol of psi, right? Psi W representing the water potential, right? Psi W of pure water is 0. That is a question multiple times this have been asked. It's a very old question. So what is the water potential of the pure water? 0, right? So what will happen? What is the water potential? Actually, the water potential is the amount of free energy or free kinetic energy available in the water. That is called water potential, right? So what will happen? The water molecule have kinetic energy, lots of kinetic energy, maximum free energy, right? When you add the solute molecule like this glucose. So what is this glucose? The glucose is solute molecule. These solute molecule, they reduces the free energy of water molecule because they collide with this one, and due to collision the loss of energy takes place. So maximum free energy of any water is called the water potential, and that is 0. When we mix the solute molecule, the water potential will decrease. So what will be the water potential of this solution? The water potential of solution will be always what? Negative. Is that clear or not? Clear? Is that clear or not? WS is called solute potential. Solute potential is that amount of the energy which reduces due to addition of the solute, right? And what is the pressure potential? Pressure potential is the amount of the pressure, right? The amount of the pressure which is imparted by the water molecule on the membrane. That is called pressure potential. So water potential, for there is a formula. Water potential is equal to pressure potential plus solute potential. Is this formula clear? Should I ask question? No reaction, not at all. Nobody is reacting. Mamuna, what happened to you? Iba, Aksa, why you people are not reacting? So I'm in fix. Either you are getting it or not, because this is not that much simple. Okay, so this is the water potential, right? The water potential is equal to pressure potential plus solute potential, right? what is the plasmolysis? This is also important for medical examination. What is plasmolysis? Can you give me any example of plasmolysis?
So red blood cell in salt solution.
Sorry, let me.
RBC in salt solution.
Yes. Very good. Good. So that is the plasmolysis. Look, now this is the normal cell, right? Now what is happening? I have told you hypertonic solution means the solution with the high concentration, right? So outside this blue color area is higher concentration, means hypertonic solution. So what will happen when you will put a cell, a plant cell in a high concentrated solution, right? So what will happen? The water will start moving from vacuole towards the outside. So when water start moving from inside to outside of the cell, this is called exosmosis. Means when the water is coming out of the cell vacuole. Basically, the water remains stored in the cell vacuole. So when water start coming out of the cell vacuole, this is called exosmosis. So this is the a normal cell where exosmosis is taking place. Means water is coming out, right? Now what will happen? The second thing is that that is the uh, plasmolysis, right? So what is the limited plasmolysis? So this first diagram is of normal cell, right? So when exosmosis takes place, we call it limited plasmolysis, limiting plasmolysis. So first limiting plasmolysis will take place, right? So what is the limiting plasmolysis? When exosmosis start taking and cells start becoming loose and the water is coming out from the vacuole. This is called limiting plasmolysis. Then second stage is incipient plasmolysis. What will happen? More water will come out of the vacuole. Due to that, what will happen? This protoplasm will start leaving from the corner, right? Corner of cell wall. So the protoplasm will leave cell wall at the corner. This is called incipient plasmolysis, right? Then what will happen? The third stage, more water is coming out. When more water will come out, this protoplasm will remain connected with the cell wall or cell membrane, at some places, and at most of the places, this protoplasm will leave the cell wall, and the hypertonic solution, hypertonic solution will get filled in this area. You can see the yellow area. The water from outside or solution from the outside get filled in this area. This is called evident plasmolysis. And there is a final plasmolysis. In final plasmolysis, the protoplasm will become of round shape and it will leave the cell wall from all the end. The connection between the cell wall and the protoplasm will not be over there. This is called final plasmolysis, right? Is it a reversible process or not? Tell me. That is my question. Is it a reversible process? Can we reverse it? Can we reverse it?
Yes, sir.
Eva? Yes. Is it possible? Ellen?
Yes, sir, it is.
Okay. Yes. Very good. So, when it's possible, is there any time limit or any time?
So when we reduce the concentration of the surrounding, right?
Yes, that is also possible. So, suppose that a cell got plasmolyzed, right, and it is kept for some time. Is it possible to make it alive again? Not possible. Look, if the plasmolysis is done and immediately after the plasmolysis you place the cell in the water, that will get reversed, but after some time it will not be reversed, right? So the reverse process is called deplasmolysis, right? Note it down. If plasmolyzed cell, and take the screenshot of this one. If plasmolyzed cell is kept in the water immediately after, if the plasmolyzed cell kept in water immediately after the plasmolysis, it will regain its structure and shape, and the process will be called deplasmolysis, right? So remember all the point, right? You will get the question. You can get the question. What is evident plasmolysis? What is incipient plasmolysis? What is limiting plasmolysis? Right? And everything is clear from this diagram, right? So stages of plasmolysis, remember, right? In the normal cell, I have written in number 1, that is the limiting plasmolysis. Number 2, incipient plasmolysis. Number 3, evident plasmolysis. And the 4th one is final plasmolysis. Okay. Water absorbing system, right? So apoplastic and symplastic pathway. Physiology is physiology is very important. You will get at least 10 questions from the plant physiology part. 10 questions from the plant physiology. Means the weightage is 40 marks. And you will get again 10 questions from the animal physiology as well. The number of the chapter and the plant physiologies are lesser, right? Look, this is this one. First, come to here. This is a structure of root, transverse section of the root, right? So outside of the root, you can see outside of the root, this is this layer. This is called epidermis. This is called epidermal layer, epidermis, right? And outside of the epidermis, these are called root hair. Root hair. These are called root hair. Now, after there is a cortex. This is endodermis. This is C, right? Vascular bundle, right? Xylem and phloem. These are the xylem which are meant for transport of the water. Now you can see here the water enter inside the root here and entered inside the protoplasm, means vacuole. From this vacuole, with the help of plasmodesmata, it entered into the cortex. In the cortex, from one cell to another cell to another cell. So then in the cortex, from the cortex, it entered into endodermis, right? It entered into endodermis. From endodermis, it entered into the xylem, right, at the final destination. So when water start moving from inside the cell, water start moving, water take the pathway inside the cell, right? This is called symplastic pathway. Means from one protoplant to another, to another, to another, to another protoplant. This is called symplastic pathway, right? The second thing is that here the water is moving between the protoplasm and the cell, right? Water is moving between the protoplasm and cell, right? Means the space between the protoplasm and cell wall, the water is moving with that space. Means it is not entering inside the protoplasm, right? You can see when the water do not enter inside the protoplasm, it move outside of the protoplasm. This is called apoplastic path, right? But in the endodermis cell, there is a one thing. Endodermis cell, there is a Casparian strip. You can see this is a Casparian strip. Caspian strip is impermeable to water. That is impermeable to water, right? There are certain pores or there are certain gaps between the Caspian strip. The water can move through there only, right? So the apoplastic pathway. So anyhow, the water either coming through the apoplastic pathway or symplastic pathway, water have to enter inside the protoplasm of the Caspian endodermis, and means here it have to follow symplastic pathway. In the endodermis, it have to follow the symplastic pathway. Apoplastic pathway is not possible in the endoderm region. The rest of the cell, it may be symplastic or apoplastic, right? Then it goes to the vascular bundle. Okay. Now the same diagram have been shown in above as a schematic diagram. Look here, the first one between the this is the cell wall. This is the cell wall. You can see this is the cell wall. This is the plasma membrane. Cell membrane means plasma membrane, right? Here you can see the water is moving between the cell wall and the plasma membrane in this space, and water is moving from here to here. Now, at this point where the endodermal cell, there is a Caspian strip. So water cannot cross to the apoplastic pathway here. Here the water will come to the symplastic pathway and will go on through the symplastic pathway. Before here, the symplastic and apoplastic both pathway are possible. Now in the symplastic pathway, the second number is symplastic pathway. Water entered into inside the cell wall, right? And then it entered into cell wall of the this cell, and then it will enter into the vacuole. From one vacuole inside, it will move to the another vacuole with the help of plasmodesmata. Here, the water is moving through, taking the inside pathway, inner pathway. So this is the called symplastic pathway. But again, the endodermis, the apoplastic pathway is not possible because of the Caspian strip, right? And this is the good. In symplastic pathway, the of like those things, virus, bacteria, those small organisms, mainly virus, they get filtered over here, right? So that's why apoplastic pathway is not that much preferred. Symplastic pathway is very important. Hope it should be clear to all of you. Clear? Is that clear? LM, is it clear? What is mycorrhiza? Tell me. What is mycorrhiza? Important question. It has been asked. Aksa, what is mycorrhiza? Hiba, I am not listening you. Are you there? I think your microphone is not on. Because you have switch on, but it got your sound. Okay, no issue. So what is mycorrhiza? Any one of you, please tell me. Aksa, any idea? What is mycorrhiza? No idea? Please try to understand. Basically, the gymnosperm, they are gymnosperm plant. They are basically in the higher plant. Fungi make a cover, like a covering, around the root of the gymnosperm. So what it do? It help in the absorbing the water, right? So fungi take the water and mineral. Fungi provide the water and mineral to the root. In return, the roots are providing food to the fungi, right? So both are helping each other. So this kind of the relationship is called symbiotic relationship, right? We have studied in ecology symbiotic relationship. So mycorrhiza is a structure where gymnosperm root and fungi, they are found in the symbiotic stage, and they help in the water absorption, better water absorption. Why? Because the roots are not that much. If efficient in absorbing water. So this is called this structure is called mycorrhiza basically. Okay, mycorrhiza is also important. Now come to the transpiration part. What is the transpiration? So the transpiration we are studying since class 9th. So why look, root is absorbing water from the soil. They absorb the water from the soil. Then the stem drop the water. Then water goes up. Then what happens? This is the lower surface that is called abaxial surface. The lower surface of the leaf, they have higher stomata, higher number of stomata. So number of stomata are more in the upper end of the lower end of the leaf. So lower end is called abaxial surface, right? So in abaxial surface, the number of stomata are more, and the upper surface of the leaf, that is called adaxial surface. In adaxial surface, the number of stomata are low. Why? Because the light fall directly on the upper surface, right? If there will be more stomata, there will be more loss of the water. To minimize the loss of the water, to minimize the loss of the water, to minimize the loss of the water, there are more stomata on lower surface and there are less stomata on the upper surface. Now look, the unused water evaporate from the leaf surface. 98 to 99% of the water is lost through the transpiration. Only 0.2% is used in the photosynthesis and 0.8 is used in the plant growth. So my question is that water is very precious for the plant. Water is very important for the plant. If this water is very important for and the plant work very hard to absorb the water. If water is that much important and plant is working hard to absorb the water, why they lose the 90 to 98 to 99% of the water? Or why the transpiration takes place? What is the purpose behind the transpiration? Come on, guys, it's a very simple and logical question. What is the purpose behind the transpiration?
So to regulate the temperature.
Very good, excellent. They it helped to release the temperature, right? It helped to release the temperature. So they help in releasing temperature. That is the important. Now, what is the guttation? So plant have to maintain their. Remember this ratio. This ratio is very important. Now come to over here. What is the guttation? It is very important. There are some plant, and these plant are basically found in. You can see. This is the picture of garden nosturium. It's a plant, and these are the water droplet. They are the water droplet. These are the water droplet, right? This is the water droplet, right? Water droplet. So these water droplets are getting accumulated, right? Garden nosturium, right? So its guttation is taking place. So basically, those areas where the daytime are very hot and the nights are colder. In those areas, the guttation basically takes place. For guttation is the water is coming out of the leaves in the form of droplet. That is called guttation, and it's very fast in South Africa and all those countries where the temperature is very high in the daytime and low in the night time. Right. So what happened? There are pores, right? Pores at the tip of the or margin of the leaves. These pores are known as hydatodes, right? Hydatodes. So these hydatodes are a pore from where the water come out in the form of the droplet, right? Basically, they take place where the day temperature is very high and nights are cooler, right? So the example of the guttation, it takes place in oats, it takes place in tomato, cucurbita, right? This is the diagram of uh, hydathode. The outer side of the hydathode is guarded by the guard cell. There are some loose cell epitheme. This question has been asked. There are some loose cell at the end of the hydathode. These are called epitheme. Epitheme are basically loose parenchymatous cell, and these are the xylem tracheids which supply the water. Right? Is that clear or not? Guttation. Clear?
Yes, sir.
Okay. It's a very interesting. What happened in South Africa? There is a tribe that is called Masai. Masai are found in African continent. So what Masai do in African continent? Masai have they collect the ostrich eggshells. They make its bowl, right? They use it bowl like. So what happened? There are large leaf trees where the guttation takes place frequently and very speedy manner. So at the evening time, they go and they put these bowl, right? These bowl which are made up of the ostrich shell, and at the margin of the leaves. And when the at the morning when they reaches, these bowl get filled with the guttation, and they collect that water and they use this water for drinking purpose whole day, right? So that is the use of the social use of the uh, or human use of the guttation water. Look, there is a theory of water. This theory is called transpiration pull theory, right? Transpiration pull theory. Transpiration pull theory was given by uh, two scientists, Dolly and uh, Dixon and Jolly. Sorry, Dixon and Jolly. So Dixon and Jolly are two scientists who have given this theory of transpiration pull theory, and the same theory is also known as cohesion tension theory. Right? Look, this is the diagram. So you have to remember what is the transpiration pull theory or what is the cohesion tension theory, and the same. So both are the same name, and who given it? Dixon and Jolly given this. Now let me explain you one by one. Look, uh, this come to this diagram first. This diagram. So you must have seen that when we uh, drink the cola drinks, what we do? We take that straw. We put that straw in the cola drink, and after that we start sucking it. Right? When I start squeezing it or sucking it, so then what happened? We put a pressure, right? A suction pressure. So due to that suction pressure, the cold drink starts raising, right? So that's how we drink the cold drink. In the same way, these xylem tubulate work as the straw of the cold drink, right? And the pressure, that suction pressure, is created by transpiration pull, right? So the same thing is happening. Look, this is a capillary. Transpiration is pulling it, right? Water molecule. So when the transpiration pull the water molecule, these water molecule work like a chain. They transfer the pressure like a chain, right? When this water molecule is pulled, then this also pull. It also get pulled. It also get pulled. It also get pulled, and it also get pulled, right? So the strain which is created over here, the strain which is created over here, that pull the water. So the water molecule attract each other with the help of a kind of the a force of attraction. This is called cohesive force. So cohesive force is the type of the force which is found, right? So a water molecule attract another water molecule with the help of the force that is called cohesive force, right? So cohesive force, it work between it work between same type of molecule, same type of molecule, that is called cohesive force. right? Now, this force, uh, with the help of cohesive force, water molecules make a chain-like structure. And because of making this chain-like structure, they are pulled like a string. Then the water molecule, there is a force between the water molecule and the wall of the xylem, right? This force, the water molecule is made up of different material, and wall of the xylem are made up of different material. So this force is called adhesion force. So adhesion force, it work between the different molecule, different material, right? This is the force of attraction which work between the different kind of the molecule. That is adhesive force. So that's why we call Fevikwik, Fevicol, and all that as adhesive gum, right? Why? Because they create a force between the different type of the material, right? So cohesive force, it help the pulling the water chain, and adhesive force, it prevent water to pull back, right, or fall down. The same thing is working over here. Look, this is the leaf structure. This is the upper epidermis part. These are the guard tissues, these are the parenchyma tissues. And now you can see this is the guard cell, right? The water is coming out in the form of water vapor. So a pull force is created at the mouth of the stomata. So at stomata, there is a pulling force which is created. So it pulls, right? When it pulls, there is this one xylem, right? There is a water carrying tubes. They're called xylem tubes, and these xylem tubes, the water start rising inside these xylem tube. Inside xylem tube, there these two forces work: cohesive and adhesive force. Cohesive force, it help in the pulling the water column, and adhesive force do not prevent the fall down of the water column, right? And that's how the water reaches to the top of the plant. And this theory has been explained by Dixon and Dolly. This is called transpiration pull theory. So transpiration start a pulling force, or start putting a pulling force, right? And cohesion do not allow it to fall back. And these xylem work as the capillary, and this transpiration uh, work as a uh, 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 transpiration pull force. It create pull force. Is that clear?
Yes, sir.
Transpiration pull theory. Okay. Please take this screenshot. One more thing. This to explain, a continuous water column is maintained by cohesion and adhesion force. Cohesion or tensile force, uh, uh, it make the water column, and transpiration create a pull, and that's how the water is reaches at the top of the plant. Please take the screenshot of this one. It is very important. Look, how does the transport of mineral nutrient takes place? Very, very, very important question. So basically, as when I was telling you, the last class when I was teaching you the active and passive transport, the last class we discussed, I told you that the concentration of the salt is more inside the plant, right? And outside the plant, there is a low concentration. Now, if plant have to absorb more mineral, so look, what will happen? This is a root of the plant. This is the root of the plant, right? Now, let's talk about the minerals. So there is I mineral concentration, and the outside there is a soil. Soil have low mineral concentration. Now, what should be the diffusion should take place from higher to lower, but it is not taking place. Again, the ions are going inside, right? Ions are going inside. So, what will happen? These ions are moving against the concentration gradient, right? Against the concentration gradient. So, ions are moving against concentration gradient. And you know, when anything have to move against the concentration gradient, it have to, what it will do, it have to uh, spend the energy. So this is mostly active process, and uh, uh, the transport of mineral nutrient takes place by the active process and the expense of ATP. Some of them by the passive process, means some of mineral also takes place to passive process. Endodermis have Casparian strip, which is made up of the suberin, right? So Casparian strip is made up of a material, and what is the name of that material? Name of that material is suberin, right? And then chief sink for mineral is growing part of the apical and lateral part. So chief sink, what is the sink? Sink are those area where the requirement of the mineral is most maximum at the uh, plant part, right? So basically, the maximum requirement is generated at the growing part of the plant, right? So at the growing part of the plant, the maximum minerals are required. Fine. So what happens that the growing part of the plant are known as the sink for the mineral, right? Next one. Most radially metabolized substances or most radially metabolized uh, elements. So most readily metabolized element are phosphorus, which are the easily available. Phosphorus, sulfur, sodium, and potassium. Which are not remobilized, that is the calcium. What is the remobilization? What is the remobilization? Any one of you? Can you tell me what is the remobilization? Remobilization. Look, when leaves become yellow, right? It have to fall down, right? Means defoliation of leaf takes place. Many of the minerals are taken by plant body, taken by the plant system back, right? So these elements, they important elements, they come back to the plant. This is called remobilization, right? Remobilization. Okay, and most radially remobilized means the maximum remobilization takes place: phosphorus, sulfur, nitrogen, and potassium. Which is not remobilized, this is the calcium which is not remobilized. It do not come back. This question have been asked many times. Tell me, is it clear or not? It is very, very, very important. All the points which is written over here are very, very, very important. Clear?
Yes, sir.
Great. Transport and phloem. Look. So when we talk about the transport of the food material, right? Transport of the means sugar. So transport of the food takes place through the phloem, and from where it takes place from the leaf to the root part, right? So the transportation. So when we talk about the water transport, then we that transpiration pull theory, that theory work. When we talk about the transport of the food material, the transport of the food material, basically this theory work, right? And this is called pressure flow theory or mass flow hypothesis, right? Pressure flow hypothesis or mass flow hypothesis were given by a scientist, and name of that scientist is the Munch, right? So Munch given this. Look, now try to understand how does this transport takes place. Just a minute. It is very, very, very important. This diagram will explain each and everything. What happened? This is the source or leaf cell, right? So these are the leaf cell. Formation of food takes place in the form of glucose. Glucose is a type of monosaccharide, right? So glucose is a type of monosaccharide. Further, glucose is converted into the glucose is converted into sucrose. So glucose is a type of monosaccharide. This glucose convert into disaccharide and sucrose, right? It converted into disaccharide inside the cell. Now the transport of glucose do not take place. Transport of this sucrose takes place. The first question, right? And sucrose is a type of disaccharide, right? Sucrose are formed in the green leaves on in the form of food, right? Now they are called source, right? So source, what are the source? Source are those cells where they are the green leaf, basically green leaves or the green cells, and the food formation takes place at the source, right? What is a sink? Sink is those areas where food is stored, right? Fruit, root, non-photosynthetic part, all those they work like sink, right? So formation of the food takes place at the source and it goes to the sink. So what happened? This is food is formed in the green leaves, then sucrose is converted, glucose is converted to sucrose. Now the transport of sucrose takes place from the source or the leaf cell to the companion cell. So companion cell are the cells of which one? They are the cells of phloem, right? So movement of why? Because the concentration of sugar or sucrose is more in the source leaf in the companion cell. So movement will take place. So that is a simple process of osmosis or diffusion, right? That takes place through plasmodesmata. When they reaches at the here, uh, this one, companion cell, right? So companion cell or these cells are called sieve tube, right? Now, what will happen? The movement of this food material, sugar, will take place by the active process. ATP will be expanded, and the sucrose will move from the companion cell to the this one, this cells. So these cells are called what we call these cells? These cells are called sieve tube element, right? So sieve tube element. This process is called loading, right? So what is the loading? Loading is the process in which movement of the food or sucrose takes place between companion cell and sieve tube element. And this is the active process, and at this process, the expense of energy takes place. Now, this ATP breakdown and this sucrose is loaded here in sieve tube. When sucrose concentration increases in sieve tube, what happens? Water potential become low, and water potential is high at the nearby xylem. Then water will start moving from xylem to the phloem. The water will move. Due to this water movement, now the solution formation will take place over here. Due to the high concentrated solution, the sugar start moving from upside to downside, right? Toward the sink process, right? And translocation takes place the direction of concentration gradient, means higher concentration to the low concentration. Now when it will reach to the sink, right? What will happen in the sink process? Again the ATP will be spent, right, by the active process. This is also the active process. By the active process the unloading is done. So the again it is taken back by the companion cell uh, from the sieve tube element, right? This process called here the ATP is utilized, and this is the uh, unloading process, and this is the active process. Now from here what will happen? This sucrose will be converted into starch, which is insoluble to water. Due to this, what will happen? The water will again go back to whom? Water will go back to the nearby xylem, and water, whatever have been taken over here, it is reversed back. Now, what will happen? This sugar is loaded or stored at the sink part, which is called the root. Here, the sugar is stored in the form of starch, which is a type of the polysaccharide, right? So this is the loading and unloading process, where the expense of energy takes place, where energy is not required, and how does it take place? This complete process very important. Is it clear to all of you or not? Tell me. Translocation of food. It is not only important, it's a very, very, very important. Hiba, is it clear? Mamuna? Clear?
Yes, sir.
Very good. So remember loading process, remember unloading process, and this is active process or passive process. These kind of the question have already been asked, right? So this was the last topic of this complete part of the uh, transport and plant. Go through with the NCRT. In case if you have any question, ask me right now. Then I will start with the another topic. Go through with your NCRT. You have 2-3 minutes to go through with it. Any doubt? Any question?
No, sir.
No. Next one is the photosynthesis. Next is photosynthesis. Fine. What is photosynthesis? I don't think so that I need to explain that, but you have listened about the photosynthesis, but you must have, I don't think so that you have listened. There are two kinds of the photosynthesis. So basically, there are two types of the photosynthesis. Normally, the purpose behind photosynthesis is to make the food, right? So now I have question: what is the byproduct of photosynthesis? Any one of you, tell me. The reaction of photosynthesis in on the screen in front of you. Tell me the question. My question is, what is the byproduct in the photosynthesis? What is the byproduct of photosynthesis?
Heat.
What is that? Tell me, please. What you said? What is byproduct of photosynthesis? No one? Oxygen is a byproduct of photosynthesis. Look, glucose is the product, oxygen is byproduct. Then what is the water? That question I've been asked. This both question I've been asked. What is the water in reaction of photosynthesis? Write it down. What is waste product? Write down. In photosynthesis, glucose is product, oxygen is byproduct, and water is waste product. And water is waste product. Look, photosynthesis can be of two types: oxygenic photosynthesis and anoxygenic photosynthesis. Oxygenic photosynthesis, oxygen is evolved. It is written over here. This basically takes place in blue-green algae and green bacteria. In these two organisms, the photosynthesis takes place. That is the oxygenic photosynthesis. What happened in oxygenic photosynthesis? Look, this carbon dioxide reacts with 6 CO2 plus 12 12 molecule of the carbon dioxide. Now, in the presence of light and chlorophyll, and in the presence of enzyme, formation of glucose takes place. Then oxygen is evolved, and water formation also takes place. Right here, the oxygen which we breathe, from where it comes? From water or from carbon dioxide? Tell me. Tell me, guys. Tell me, guys. No, the oxygen which we breathe, that comes from the water, not from the carbon dioxide. Right? If you want, please note it down. So oxygen which we breathe and which is present in the air, it comes from the water, not from the carbon dioxide. So look, and this hydrogen is utilized here, right? Formation of, and the oxygen comes from here, right? This oxygen, it makes the oxygen from the water, it makes the O2, right? Now, in oxygenic photosynthesis, formation of oxygen is taking place. There is another kind of the photosynthesis, that is the anoxygenic photosynthesis. In anoxygenic photosynthesis, the formation of oxygen do not takes place. So, anoxygenic photosynthesis, takes place in the purple sulfur bacteria, right? 6 CO2, 12 H2S. Here in place of the water, right? Here in place of the water, a hydrogen sulfide is utilized, right? So hydrogen donor is hydrogen sulfide, 8,6C6H12O6, and then 12S and 6H2, right? So here formation of sulfur will take place in place of the water. So this is the two type of the photosynthesis. There are some factual things. 70,000 to 80,000 ton of carbon dioxide is fixed annually. Every year in environment, 70 to 80 billion ton carbon dioxide is fixed by the plant. When I say plant, it is fixed by the both marine as well as the terrestrial plant. It produces over the 170 billion ton of the dry organic matter, right? So 170 billion ton dry organic matter is plant material, right? Older estimate is that 10% of the dry matter is produced by the land and 90% by the ocean, right? But at present, the 68% of the dry matter, means the food, is produced by the land. This is the recent, right? Approximately 0.2% of the solar energy that is received by the earth is utilized in the photosynthesis. So only this 0.2% of the light, right, it is running the whole biosphere. It is running the whole basket, right? Photosynthesis. In photosynthesis, only 0.2% of the solar energy is received by the Earth's surface is used in the photosynthesis. Now come to here. These the name of these scientists is also important, right? Joseph Priestley, they said that the plant have capability. He discovered the oxygen as well as Joseph Priestley was the person who discovered O2, right? Then Joseph Priestley said that plant have property to convert foul air into pure air. For foul air, he used the term phlogiston, and for pure air, he used dephlogiston, right? The phlogiston and dephlogiston have been asked. This question has been, what is the dephlogiston? That was the question. Dephlogiston, he used it for the what? Oxygen, and phlogiston he used for the carbon dioxide. Is that clear? So Lavoisier was another scientist. Lavoisier said that phlogiston is carbon dioxide and dephlogiston is O2. Please take the screenshot of this one. This is structure of chloroplast. We'll again study it in the this one cell biology part, right? We'll again go through with it in the cell biology part, one more chapter. So chloroplast is a site for photosynthesis, where the photosynthesis takes place. The chloroplast is made up of two membranes. Some of the scientists says that it is made up of three membranes. This outer membrane, this inner membrane, and this third structure. These granum are considered as a third membrane holding, right? So, but most of the scientists, they have the uh, unanimity and consensus that it is made up of the two layer only, right? So look, this is outer structure, this is inner structure, means inner membrane, and inside there is a gel-like substance that is called stroma. Inside the stroma, there are certain structures. These structures are called grana, and a single structure is called grana, right? Look, now what happened that light falls on the grana, then photolysis of water takes place. That is called Hill reaction. Photolysis means photo means light and lysis means breakdown. Photolysis is the breakdown of the water in the presence of light. This is called photolysis, right? So photolysis of water takes place. This is also known as the Hill reaction. Hill reaction, right? So Hill reaction or photolysis of the water or light reaction takes place in the granum. Then what happened in this stroma? The dark reaction, that is CO2 fixation, takes place in the stroma. So half of the reaction takes place here and half of the reaction takes place here, right? Look, in the leaves, the mesophyll cells have the chloroplast, and these chloroplasts are found at the outer side, means outer side of the cell, so that they could receive the maximum light, right? Again, this structure is called granum. This pile, this complete set of the pile, this is called granum, right? And collectively they are called grana. Each grana is made up of a tablet-like kind of the things. You can see these are looking like choco pie. So this single structure, right? This single structure is called thylakoid. So every grana is made up of granum is made up of number of the thylakoid, right? So thylakoid, this is the structure of the thylakoid. At the wall of thylakoid, these are the walls of thylakoid. At the wall of thylakoid, there are chlorophyll molecules, right? chlorophyll molecule which are found. These chlorophyll molecule are made up of magnesium. That's why we can say they are the magnesium compound. And the this outer part, this outer side part, this is called non-appressed part, right? And these part where the two thylakoid come in contact with each other, these are called the appressed part, right? So at these are the non-appressed part and these are called appressed part. Photolysis of water here. Photolysis of water takes place at the appressed part, right? When at the appressed, what is the appressed part? Where the two thylakoid are in contact with each other, their surfaces meeting with each other. So this point, which are in contact with another thylakoid, this is called appressed part. In the appressed part only, breakdown of the water molecule takes place. That is the photolysis of water molecule takes place. Look, this is the 2H2. This water molecule breaks down and formation of 4 hydrogen oxygen molecule and formation of 4 electron takes place. This process called photolysis of the water, right? Tell me guys, is it clear or not? Is it clear to all of you? Is that clear?
Yes.
Okay. Now come to the complete mechanism of photosynthesis. Look. At the daytime, what happened? Light falls on the grana. Water molecule breaks down, right? This is called photolysis reaction or the water light reaction. Formation of oxygen takes place. In light reaction, ATP is produced, this one, and NADPH is produced. Both are the energy currency. Look, here I have told that 1 GTP is equal to 1 ATP, 1 NADPH is equal to 3 ATP, and 1 FADH is equal to 2 ATP, right? So here ATP formation takes place, means energy is formed, NADH is formed, means energy is formed, and then this energy is utilized in the Kelvin cycle. What is the Kelvin cycle? Kelvin cycle, it goes on day and night, right? So we can say this reaction which is taking place at the daytime, this is called light reaction. So light reaction takes place in the presence of light. That's why we call it light reaction, right? So this is the light reaction. Light reaction can take place at the daytime. And another is called dark reaction. Dark reaction takes place at the day night only, all time. This light reaction provide the ATP and NADPH to dark reaction, and then what it do? It fix the carbon dioxide from environment and convert it into the, and then again give back to the this NADP plus and ADP plus. It is again given back to this one and will convert into NADPH and P. Right? Is that clear? So that process is clear. So in light reaction, you can say oxygen is produced, NADPH2 is produced, ATP is produced. And in dark reaction, food is produced, sugar is produced. Is it clear? Clear, guys?
Yes.
This equation, remember this comparison, right? We will require it in the photosynthesis again, and even in the that respiration part. Photosystem, right? Photosystem are nothing but just they are made up of the chlorophyll molecule. Like this is the photosystem. This is the structure of photosystem. So photosystem is made up of two things: a reaction center and antenna molecule, right? So these are the reaction center and these are the antenna molecules, right? So look, the reaction center is made up of two things: a chlorophyll A molecule. This is a special chlorophyll A molecule and a primary electron acceptor, which will accept the electron. Two things make the reaction center. Around this reaction center, there are number of the small molecule. They are called antenna pigment. They are basically made up of carotenoid. So what happened? They capture this small frequency of the light photon, right? They all, like you can see the diagram, they capture, and finally they transfer their energy to reaction center. This, when this reaction center will collect the energy, it will give energy further. That's how the photosystem works, right? There are two photosystems. So these antenna pigment are made up of some molecule like carotenoid molecule, right? And this reaction center is made up of basically chlorophyll A molecule, right? Now there are two photosystems, photosystem 1 and photosystem 2. PS1 is called P700 and PS2 is called P680, right? Why the 700 is called first? Because it has been discovered first, and P650 discovered later. That's why it is called PS2. I don't get confused. Now you can see that PS1, photosystem 1, is found at the appress part, right? And photosystem first, it is found in non-appress part. Look, in photosystem second, this is the oxygen evolving complex is found where the water movement takes place. Water breakdown takes place, and due to the breakdown of the water, oxygen is produced, right? So this is the reaction center. Now reaction center 2, where the reaction center 2? Reaction center 2, you can see, reaction center 1. Reaction center 1 or photosystem 1, they are found at the non-appress part, right? And in the same way, there are ATP synthase enzymes. Look, photosystem 1 is made up of, it is found at the non-appress part. It will have more chlorophyll A molecule, chlorophyll B and carotenoid is found in the less amount. This is, it is also called LHC1, and this is the photocenter, this one, photocenter or reaction center, right? The reaction center is made up of chlorophyll A molecule and P700, right? Let me write it down more clearly. The reaction center. Reaction center is made up of chlorophyll A molecule and P700. Both cyclic and non-cyclic photophosphorylation. We'll discuss what is the cyclic, what is the non-cyclic phosphorylation. They take part in both cyclic and non-cyclic both. Right. Now talk about the photosystem second. Photosystem second is located in the non-appress part. We have seen that it has chlorophyll A molecule, chlorophyll B molecule, and carotenoid molecule. This is very important. The questions have already been asked. Now it have photocenter, oxygen evolving complex, right, which help in the oxygen evolution, LHC, light harvesting complex second, right. Photocenter is made up of a dimer of chlorophyll A molecule. Dimer means there are two A chlorophyll molecule, chlorophyll A molecule. It have oxygen evolving complex, right, that is made up of magnesium, calcium, and chloride. It help in the evolution of oxygen. Oxygen evolving complex, it releases the electron that is received by the electron receptor, right? So this is the composition of photosystem second. Please take the screenshot of both photosystem second and photosystem 1 and remember it. Is that clear, Mamuna, Hiba, Hanifa, Alan, and Aksa? Tell me. And take the
Yes,
screenshot
sir.
of this one as well. Remember the photosystem 1, photosystem second. Go through with it, whatever we have studied today, and ask me if you have any question. Please. All clear? Mamuna, clear? Hiba, what's about you? So see you in next class, right? And in next class, we'll start from the photosynthesis onward, right?